Hi everyone, and welcome to this virtual chemistry lesson, where we will talk about acid and base titrations. For those of you who stumble onto this video, I'll have you know that this is gonna be about the very basics of acid and base titrations. Therefore, we won't go into monoprotic acids of any kind, nor will we go into titrations of weak acids and bases. So this will be titrations with strong acids and sodium hydroxide for starters. But for those of you who are new to acid-base titrations, welcome, and I hope you learn a lot through this video. So, titration. Well, that's one of the most common analytical techniques we use in chemistry. It doesn't matter which commercial lab you will travel to and explore, you will find that it's very common to do various types of titrations. These are not limited to acids and bases. There are such things as, for example, redox titrations, but in this case we're only going to go into the basics of acid and base titrations. The principle behind acid and base titrations is that of neutralization. When acids react with bases, they neutralize each other to form water and a salt. And we can use this to figure out unknown concentrations of either the acid or the base. There are two common methods when we do acid and base titrations. Either you do titration together with something called a pH indicator, or you do titration with a pH meter. We're going to go into both of them. But first and foremost, this is a experimental technique. So I'm going to go through what materials you need in order to make a titration. Well, first and foremost, you're going to need a burette, which is a sort of glass tube with a valve where you can close and open and therefore control the output of a solution out of this thing. It's also graduated, as you can read very clearly how many milliliters you've emptied from this. We need to hang this burette up onto something, and therefore we use a retort stand with a burette clamp. It's a special clamp that is made for burettes, pretty much, to easily attach them. There are other ways to clamp these on, of course, but a common way is using a dedicated burette clamp. A stir bar. That's something we lazy chemists like to use. It's a small magnet coated with a non-reactive material, which we then can put into our solution. Since it won't react with our solution, we're safe. Then we use a device that's called a magnetic stirrer, which we have here on the bottom of this picture. It also contains a magnet inside, and we can cause the magnet inside of this device to spin. If it does that, it will pull this stir bar with it and causing that to spin. And if the stir bar spins, we will mix the solution continuously. So it's a way for us lazy chemists to mix our solutions continuously without having to use a glass rod or something like that. In the example we use on this picture here, we use an indicator. We add a substance such as B2B to give a nice color to the solution. And I will talk about why that is right now. So pH indicators are substances that changes their color dependent on the pH of a solution. These can be found in nature quite a lot. For example, red cabbage will have different colors dependent on the pH, and so will blueberries. Quite a lot of flowers change the color of their petals dependent on if they're in alkaline or in acidic soil, and so on and so forth. And in the beginning of pH indicator history, we used something called litmus paper, which is based on a lichen that we can find and extract this a certain substance from, and then we can let that solution dry into paper, and we have litmus paper, which we can then dip into any solution and we'll get a color dependent on the pH. The most common analytical indicator, or indicator that we use in the lab, is bromothymol blue, BTB, which we have a picture of here to the right. You can see that at low pH values we have a yellow solution, and at high pH values we have a blue solution. At pH 7 we have a green solution, and that is because we have a mixture of both this yellow part and this blue part. We have a little bit of both, because at pH 7 we have both acidic things and basic things. Basic substances and acidic substances. And since we have both, we both have oxonium and hydroxide here, we get a mixture of the blue and yellow color. And you're supposed to know this color scale for BTB: yellow, green, blue. So let's go through an example of how we would use a titration to solve a problem. Say that we fill this e-flask on the picture. You don't need to use an e-flask, but it usually limits how much it splashes when you do this. Sometimes the drop can cause the surface of the solution to splash up and you lose a drop of solution. The thin neck prevents this to happen. But yeah, we can use a beaker, some other things for this. Since we have an acid, hydrochloric acid of unknown concentration in the e-flask, if we add BTB, then that will have a nice yellow color. And I want you to imagine that this picture shows sort of after the titration experiment. But once we add BTB to a hydrochloric acid solution, we'll get a yellow color because it's acidic. Then we fill the burette that we have here with 50 milliliters is a common volume that we use. There are larger and smaller burettes, but I think the standard is 50 milliliters. With a sodium hydroxide solution. So we fill this up with sodium hydroxide, and we know the concentration of that base, that sodium hydroxide. It's 2.0 molar. Then we turn on the magnetic stir here, which causes the stir bar to spin and mix this solution constantly. Then we slowly open the valve just a tiny bit, so it drops from the end of this very slowly here. Eventually, the solution here will turn green. And just when it turns green, you will stop. And that's good, because then we're at a neutral pH, and that means something in this example. If you take a little bit too much, and it's enough with just taking one additional drop here, one additional drop beyond the green color will cause this to turn blue. So we can imagine that this picture shows us when we've added too much sodium hydroxide, so to speak. So let's say that we added 15 milliliters of sodium hydroxide solution with known concentration down into the e-flask with acid of unknown concentration, and the color shifted to green just at 15 milliliters. Now we have everything we need to determine the concentration of this acid. So let's get into the details of this a little bit. So we added 50 milliliters of sodium hydroxide. If the BTB turned green, this means that we have a neutral solution, pH 7. That means that we don't have an excess of either oxonium ions nor hydroxide ions. So this will be like neutral water, which you know by now, hopefully, if you've gone into any details of acid and base theory anyway. I'm sort of making this for my own students here. So, but yeah, if it's neutral, then we have equal amounts of oxonium ions and hydroxide ions. If we have more of the oxonium ions, we will have a acidic solution, and if we have more of the hydroxide solution ions, then we will have a basic solution. But when we have equal amounts, then we have a neutral solution. This, in a titration like this, we call this specific point, we call that an equivalence point. When we have equivalent amounts of oxonium and hydroxide, This is the equivalence point, and this is what we need to use to solve our problem. That's why we want to stop the titration once we've reached the equivalence point. So, you know that you added 50 milliliters of 2 molar sodium hydroxide solution. You could make a table like this, and we've gone through how to make them before, but you just write the chemical or the formula for what happens, this physical change in this case. You write volume, concentration, and then you can calculate the number of moles. That's the product of the volume and concentration. Since you know that the molar ratio is 1 to 1 to 1, you can predict the exact number of hydroxide particles freed out into this solution. Good. So we know the number of hydroxide ions in this solution because we have volume and we have concentration. So we know that at the equivalence point, the amount of oxonium ions equals the amount of hydroxide ions, right? That means that the amount of oxonium ions will be the same as that of the hydroxide ions we got from the sodium hydroxide. So now we can figure out the concentration of our acid based on the formula you can see down here. You write the formula for what happens when you put hydrochloric acid in water. Oxonium and chloride ions are produced. Make a table. And then the thing we know from the titration is that the number of oxonium ions is 3.0 times 10 raised to the power minus 2 moles, which is given over here. Okay. You also know the ratio here. It's 1 to 1 to 1 to 1. I appear to have forgotten a 1 here, but that doesn't matter in this case. But yeah, it's 1 to 1 to 1 to 1 all the way here. That means that we need to have the same amount of hydrochloric acid molecules when we started than that we had of the oxonium ions that were produced. Because for each one of these that reacts, we will form one of those. So if we form 20 of those, we will have 20 hydrochloric acid molecules. In this case, it's way more than that. You don't know the concentration of the hydrochloric acid, but you do know that you took 50 milliliters, which in scientific notation we can write as 5.0 times 10 raised to the power minus 2. Now you have 2 out of 3 things here. We have volume and amount. Then we can quite easily calculate concentration. And that's just taking the number of moles divided by the volume. And that will be 1.5 times 10 raised to the power minus 3. So this is the concentration of the original acid that we used. So using an experimental procedure like this, we can figure out unknown concentrations of either acids or bases. We could also work the other way around. If we had a base of unknown concentration, we could have added acid with a known concentration. We would have found the equivalence point when the solution was neutral. Then this expression would still apply. And then you can use that to figure out how many hydroxide were formed in the reaction and then calculate back to the concentration of the original base, for example. So it doesn't matter which way we use. We can solve problems involving finding concentrations of acids or bases. Okay, we could also do acid and base titrations with a so-called pH meter, which is a type of voltage meter. The pH meter has a probe which we can dip into our solution of either acid or base, and it reads then the pH of that solution dependent on voltage. And you don't need to know how this works, but for now you can accept that we have a device that can directly measure pH. Many of you may have seen that. If we have this into our beaker, for example, remember previously we had a e-flask, here we have a beaker, it doesn't matter. We empty the burette with, in this case, base to determine the acid down here. And you continuously measure pH. Well, when this machine over here, represented by a nice square, When that machine shows you the pH value of 7, that's when you stop. And then you know the volume you've added. It's neutral pH, so you know that the number of oxonium and hydroxide ions are equal. And then you can determine the unknown concentration. But something fun we can do with a pH meter to better understand what happens in these solutions when we're having neutralization reactions is to construct so-called titration curves. A titration curve is, you can easily build up a titration curve by adding, say, depends, but you can add a milliliter, record pH, another milliliter, record pH, and so on. So then you will collect many different values of volume with corresponding pH values. So X values and Y values for a graph. And if you were to construct a titration curve based on, and I want to stress this, based on strong acids and bases, that's when you'll have a titration curve that looks like this. If you have, for example, diprotic acids, it will look a little bit different because we have 2 protons being released. One of the processes will be weak, the other will be strong, maybe. And if you have a weak base, then it will look different like this as well. It won't be perfectly fit 7 here, where we have the equivalence point. But for now, we're going through the basics, which this is chemistry 1 after all. So we assume we only look at strong acids and bases here for now. So what you're supposed to do is to be able to interpret one of these graphs. On the y-axis we have pH, and on the x-axis we have the volume added of 0.1 molar sodium hydroxide. All right. The hint, or my tip, to solving a problem like this is to find the equivalence point. You can find the equivalence point right there where we have a pH 7. Addition of small amounts, like a drop, will drastically shoot the pH up here because you have a very steep slope of the curve here. So just a small shift in the volume will correspond to a very high decrease in the y-axis. And that's because after this point, we've neutralized all the acid. So, when we have strong acids and bases, the equivalence point lies neatly at pH 7, right here. So you want to find this, and then we use that to solve a problem. So, you then look at this point, you follow it down, and in this case, it fits neatly to 100 milliliter. You know the concentration of the base you've added, then you know the number of moles, and they will be equivalent to the number of moles of oxonium formed by the acid. And then you can solve problems based on that, like we've gone through before. So find the equivalence point, find the volume, you know the concentration, and build on that and solve your problem. I always recommend that you make nice tables for the reactions involved, so you keep track of all the data you have. So, an example. Say that you titrated 50 milliliters of nitric acid of unknown concentration. This is a monoprotic strong acid, and the concentration of that, it has an unknown concentration. You titrate it with sodium hydroxide with 0.1 molar, according to the curve on the right. So what was the pH of the acid in this example? Well, then we find the equivalence point. We read it down to the x-axis. It's at 100 milliliters exactly. Then we know that we needed 100 milliliters of 0.1 molar sodium hydroxide to neutralize all of the acid, according to, and then you can easily find the amount of substance of sodium hydroxide by multiplying the volume and concentration. And then we take 0.1 times 0.1, because remember, we need to use cubic decimeters here, or liters. And then you will find out that the number of moles is 1.0 times 10 raised to the power minus 2. And you can make a table to look at this. Since sodium hydroxide splits up into sodium ions and hydroxide ions into a 1:1:1 molar ratio, the number of moles of sodium hydroxide corresponds to the number of moles of hydroxide ions. And then you know that at the equivalence point, this expression is valid, that the number of hydroxide ions equaled that of the oxonium ions, so they were equal amounts and neutralized each other. And then if you wanted to calculate the concentration of the original acid, that will also be a 1:1 ratio, because this will only lose one proton and form one oxonium per molecule. But this middle part here is what we use. So then we know that the amount of oxonium is 1.0 times 10 raised to the power of -2. In the original exercise, it said that we used 50 milliliters of this acid, and we write that in cubic decimeters. Then you write it in scientific notation like this. Take the number of milliliters, divide with 1000. And then you know that if we have the amount and we have the volume, we can calculate the amount per unit volume, that is concentration. Let's do that. Let's put in these two values into this expression, and then we'll find out the concentration of the oxonium ions in there. In the exercise, we wanted to find the pH, which is a measurement of the amount of oxonium in water solutions. So then we take the negative logarithm of the oxonium ion concentration according to the formula we've seen before. In this case, it's the negative logarithm of 0.2, and that will be 0.70. So 0.70 is the original pH of that nitric acid solution that we had. And the concentration of it was 0.2 molar. So that's the end of this presentation. I hope you learned something of how to use the fact that strong acids and bases react in neat neutralization reactions. I want you to use that to solve problems on how to determine concentration of unknown samples. That's it for now, and I will see you in another video.